Hey, it's my pleasure today to be talking about spatial biology with Dr. Will Wong. Will did his MD at Harvard Medical School. After Harvard Medical School, he did his PhD work in biophysics also at Harvard with Xiaowei Chuang. Postdoc with the Broad, with Aviv Regev and Tyler Jacks. And now is running his own lab, brand new lab at Harvard MGH, and starting his own lab, doing some of the most exciting spatial biology that I've ever seen so far. And hey, it's a pleasure to see what you're doing these days, Will. Welcome.
Thank you very much for that kind introduction, Dr. Beecham, and excited to share with you kind of this journey that we've taken in collaboration with NanoString on understanding the tumor microenvironment in pancreatic cancer at resolutions we couldn't dream of a few years ago. And the story I want to share today is very much ongoing, and it's looking at, I think, one of the most neglected components of the tumor microenvironment, that is the nervous system. We know that the nervous system is there. There's many different flavors. It's not just a one-size-fits-all, but we still really are in, the infancy of this field of cancer neuroscience in terms of understanding the when, the where, the how of why nerve signals affect tumor cells and vice versa. And this story is very much inherently spatial, and as you'll see, this story evolve. We started working with geomics and got a lot of insights from there, and that very naturally led into cosmics, higher resolution, narrowing down our focus on certain candidates. And then I'll leave with a little bit of a teaser, if you will, on some of the functional data that we're now collecting. And then I'll just mention throughout the slides, I'll have some pictures up of people in my lab who've contributed to this. Of course, there's innumerable people at NanoString who've contributed to this, and just for sake of space, I have their pictures kind of at the very end in the acknowledgement slide. So this is just a little bit of history. So, I mentioned this being kind of a relatively neglected component of the tumor microenvironment, that is the nervous system. But it's not because we didn't know that there was a high density of nerves in both central nervous system as well as peripheral tumors. This is a paper that I dug up by a urologist, Dr. Hugh Young, published in 1897 in Journal of Experimental Medicine, which is still around, of course. And he was basically going through all these different peripheral tumors looking at histology, and he noticed that there was this high density of nerves in almost all the tumors he looked at. And he pontificates a little bit. He's like, I don't think this is just an accident. These nerves probably aren't just here because they were there already. There seems to be some intent, but of course he didn't have the tools at the time to really figure out what was going on. So fast forward about 100 years, and Ayala and other various kind of early cancer neuroscientists started to become interested in this question. So what Ayala did was he took some prostate cancer cells, two-dimensional culture, and plated them at a certain distance, almost like a scratch wound test, from dorsal root ganglia, right, which is a source of sensory neurons. And he, through live cell imaging and immunofluorescence, as you're seeing here, he saw this remarkable reciprocal tropism between the cancer cells and the nerves. And you can see them kind of growing towards each other. And started to study this, mostly at a phenomenologic level, but also starting to look into molecular mechanisms. And this field kind of took off over the last 10, 15 years, mostly in brain cancers. Michelle Mange, Hamza Venkatesh, Frank Winkler have done a lot of work in gliomas and looking at exactly what these interactions between nerves and tumor cells are. We're still very, very far behind in peripheral tumors. But nevertheless, the excitement around this field led Science Magazine to write this feature called War on Nerves in 2019, and really highlighting the potential importance of this element of the tumor microenvironment. So in terms of what we know, I'm going to zoom, my focus is mainly on peripheral cancers, pancreatic and other GI cancers specifically. In terms of what we know, we do know a little bit. You can see from this review from a couple years ago that the types of tumor nerve crosstalk are varied, but what we mostly understand is biochemical communication. So these can be neurotrophins like GDNF, BDNF, NGF that get co-opted by cancer cells to actually stimulate oncogenic signaling. It can also be immune modulation. That's a really hot area right now. Nervous input into draining lymph nodes for tumors. How does that, or direct input, nervous input into tumors, how does that modulate an immunosuppressive versus a more inflammatory microenvironment? We're also very interested in that. Metabolic support has come into focus recently. There's been some studies showing that serine-starved pancreatic cancer cells can actually alter their codon usage to generate more nerve growth factor and actually bring nerves in that will secrete packets of serine to them, which is pretty cool mechanism. That's been shown in vitro and in vivo. And I think perhaps most exciting, and something I won't talk about today, but is the potential for actual electrical communication between cancer cells and nerves. Yeah, maybe a year or two from now, I'll share some of the data on that. So this figure is just summarizing that when we talk about tumor nerve crosstalk, we're not just talking about cancer cells, right? Here's a pancreatic cancer cell depicted here. Here's a neuron, but there's also cancer-associated fibroblasts or pancreatic stellate cells. There's tumor-associated macrophages or MDSCs, and many other components that are part of this dialogue. And what I realized when I was looking at the literature was there was this major gap in this field. Most of the prior studies really focused on specific pathways of interest where there was already a hypothesis from prior work, so definitely not comprehensive. And then the other problem was most studies lack cell type specificity. So you get a bulk signal and you don't know when you see something differentially expressed in your setting of interest versus not, which cell type it's coming from. And that's obviously very important from a mechanism standpoint. And then lastly, and most relevant to this talk, is spatial context, right? So I didn't get into the whole story, but the reason I got interested in this field was actually the first couple pancreatic cancer patients I had as a medical student had this intractable pain that opioids didn't touch. Basically, they were completely unconscious before they would tell us they were comfortable. And that led me to talk to pathology, and then I learned about this concept, perineural invasion, where cancer cells actually invade into the perineurium, which we know is at least partially responsible for this neuropathic pain, but is only one manifestation of tumor nerve crosstalk. In any case, the thing that really bothered me from a research standpoint is that when you look at a histopathology report, when they mark perineural invasion positive or negative, it's just a single focus in a huge tumor of perineural invasion, it's positive. And if they don't see, in 10 high-powered fields, if they don't see it, it's negative. And you miss on both sides, right? So I won't get into that today, but I think that's where spatial context really matters, because even in a particular tumor, some cancer cells are engaging with nerves, whereas others aren't. And by averaging all that across a tumor, we're certainly introducing a lot of noise. So to try to fill this gap in knowledge, as I mentioned, we've had this long-standing partnership with NanoString, and we came up with a strategy that we thought would really maximize our power of trying to understand in a comprehensive spatial screen cell type specific gene expression that's associated with tumor nerve proximity. And I used proximity rather than perineural invasion, which is the more common histopathology term, because as we've seen, and as others like Nisha De Silva did some geomics work on this as well, have seen that actually cancer cells and nerves, even being close to each other but not actually physically interacting, you already start to see changes in gene expression in both cell types, and you also see major negative prognostic implications. So we designed this screen with some of my collaborators at MGH Pathology, where we generated these 3 millimeter bore TMAs, as you see down here, and then on a consecutive section, we did whole transcriptome digital spatial profiling with the geomics platform. And there's a couple unique things about this. So one, we had 12 of these, so we had well over 250 cores. The other unique thing about this was we matched regions in the same tumor. So we had about 30 tumors, and we had regions where there was tumor nerve proximity and regions where there was not. And that kind of cuts across the interpatient genetic heterogeneity. We're really trying to nail down molecularly what might be mediating tumor cells and nerves to want to be close to each other and interact. We also had some nerve control. So I'm showing the 3 different types of regions of interest, the RLIs here. So on the left here, this is a nerve control. So pink is S100B. Sometimes we can use beta-3 tubulin as another relatively nerve-specific stain. Here, there's no cancer cells around. And the area of illumination is simply just the nerve. And then in the middle here, this is what we call a nerve-positive region. So you have this pink nerve in the middle, surrounded by green pan-cytokeratin-positive cancer cells and cyan, smooth muscle actin-positive fibroblasts. And I'll get into the AOI strategy in a moment. And then lastly, we had these nerve-negative areas in the same tumors with nerve-positive and nerve control areas. And here you're seeing no pink nerves, but you see a lot of cancer cells and fibroblasts. And here we just kind of segment the conventional way. in two parts. This is a quick view of all 12 TMAs that we ran on the genomics platform. And then this is for the nerve-positive ROIs, our segmentation strategy. So we interrogated the whole transcriptome of the nerve, cancer cells within 100 microns of the nerve, within 300 microns of the nerve, and then similarly for the fibroblasts, within 100, within 300. So this gave us this kind of distance gradient information, as well as cell type specific information. As I mentioned, this was at the whole transcriptome level. So we felt like this was our best kind of highly powered chance at filling those three gaps that I mentioned earlier. So just to take a very quick step back, in some of our prior work on geomics and also with single nucleus RNA sequencing, we had refined the molecular taxonomy of pancreatic cancer. We recapitulated some transcriptional subtypes like basaloid classical, which had been known even at the bulk level, but we also identified some unique transcriptional programs like the neural-like progenitor or NERP program, which was very interesting to us because in data I'm not going to show now, we now have match data, some clinical trial data, as well as functional data where we've identified the master regulator of this program. But at the time, we were already very excited because we saw this significant enrichment in post-treatment residual disease of this neural-like progenitor program, and also in independent cohorts we saw a significant association with negative prognosis in our patients. So we were hypothesizing that somehow similar to the mesenchymal EMT program that had long been known to play a role in more invasive, aggressive, treatment-resistant disease, that this neural-like progenitor program we had newly identified may be an alternative pathway in which pancreatic cancer cells and perhaps other epithelial cancer cells use to resist treatment and cause other bad effects. So one of the first questions we did, going back to this spatial tumor nerve screen, is we scored malignant cells in areas where there was perineural invasion or tumor nerve proximity versus areas where there was not, and for each of these 7 programs that you see up here. And when we looked at all the nerves and all of our cores and ROIs, we saw that the mesenchymal program here was strongly enriched, p-value is less than 0.0001. It was strongly enriched in areas of tumor nerve proximity, and the classical program, which has the most favorable prognosis in pancreatic cancer, was significantly depleted in areas of tumor nerve proximity. We also saw the basaloid, which has some relation to the mesenchymal program, smaller magnitude, but also significant enrichment. This NERP program that I just spoke of, the neuroendocrine program as well. So there was a lot of significant hits here, but we were interested in actually parsing this out a little bit more. So we noticed right from the get-go that there were 2 very distinct patterns of tumor-nerve interaction. One we kind of call large nerves only, and it looks something like this, right? You have this large pink nerve, you can see tons of nuclei. This is clearly an intrapancreatic ganglia, something that was intrinsic to the pancreas even before the tumor formed. And you can see these green cancer cells have heavily infiltrated. I mean, this just looks painful to me looking at this. They're basically have splayed apart all these nerve fibers. The nerve is not even the recognizable bundle at this point, or a ganglia for that matter. And then you have some fibroblasts kind of interwoven and then around the outside. So when you look at this pattern of invasion only, you see there's still a very strong association with mesenchymal cancer cells and the basaloid, and then a depletion in the classical, but everything else I just mentioned, the significance goes away. If you look at this other extremely different pattern of invasion where you see this kind of speckle pattern, right? So, all these little pink dots here, there's no nuclei associated with most of them. They appear to be more like, neurites, nerve fibers, if you will. And while this is just a snapshot of human tissue, it definitely evokes the thought that this might be a little bit more of a neo-innervation pattern, if you will, where there might be active recruitment into the local microenvironment of these nerves. And when we look at this, we still get the association of mesenchymal basaloid, but now we get and classical being depleted, but now we get the NERP and the neuroendocrine back being significantly associated. So just to summarize those kind of initial findings, mesenchymal EMT, basal-like cancer cells we find associated with nerves regardless of size. This neural-like progenitor, neuroendocrine cancer cells seem to only associate with small nerves and fibers, and then classical cancer cells are relatively excluded from nerve-infiltrated areas. So a working hypothesis for us was then that EMT programs may facilitate nerve invasion, really irregardless of what the nerve structure looks like, while NERP, neuroendocrine programs, may mediate nerve recruitment and outgrowth. And you know, I'll only have time to show you kind of the second half of this hypothesis, but some of our early data looking at invasion in a functional manner suggests that, that both parts of this hypothesis are likely true. So we took a step back at this point. So we were interested in seeing things at the subtype level, but we wanted to know, you know, what specific molecular mediators are potentially generating these phenomena that I just mentioned, if our hypothesis is true. So so just focusing on the cancer cells, we did a DE analysis. So on the right here, this is enriched in nerve positive areas and then depleted on the left. And when you look at this volcano, we applied a pretty strict algorithm. We were just looking at the top 100 weighted genes from each of our programs. That's from single nucleus RNA sequencing. And we looked at the overlap with these DE hits. And what we noticed is that some of the top hits, you can see MES and BSL stands for mesenchymal basaloid. Some of the top hits were actually very EMT-like, which is not surprising. So we picked, using, again, the top 100 weighted gene cutoff, we picked the top 4. So the top 4 genes you can see here are labeled and they're scattered throughout. These green NERP genes, again, using the top 100, we took the top 4, and then there was a positive literature control and then a negative literature control. So we took those 10 hits. We also, this is just us running GSEA and looking at what are the top geo terms associated with enrichment in nerve positive cores. And you see things like cell motility, circulatory system, vascular development, collagen-containing extracellular matrix, nothing particularly surprising at that level. Another way to look at this D analysis that maps to our programs is to just, again, this is top 100, top 200 actually, sorry. Positive and negative direction, enrichment depletion. You know, you see that mesenchymal, basal, and NERP are the top 3 in terms of the most enriched for these positive DE genes. And classical, and a little bit surprisingly, our squamoid program are the most depleted. And then just to remind you, when we go back to the spatial data, we had this kind of, this built-in concentric radial strategy in terms of how we obtained our data. So we looked at these top genes, all these enriched genes, and we looked at binary comparisons between expression of each gene in the inner ring, so within 100 microns of the nerve, versus the control, outer ring versus the control, and then inner ring versus outer ring. And for each of these comparisons, we're basically seeing that for the most part, all these log-fold changes are above 0, right? There's a couple exceptions. And when we just zoom in on our top 10 candidates, you can see the negative literature control. When you go from a control region to the outer ring to the inner ring, you see kind of this more or less monotonic decrease in gene expression. And with one exception, with all of our positive hits, you see a monotonic increase in gene expression. So this was really interesting to us. But one of the issues with this is, you know, at the whole transcriptome level, we were able to do an unbiased kind of discovery, find our top 10 candidates. But without higher spatial resolution, there's obviously some potential confounding with these 100 to 300 micron rings. So that's why we were really excited when we had the opportunity to work with the TAP program and try spatial molecular imaging. We started with their 1,000-plex panel, initially without any customization. I won't belabor, I think from Dr. Beecham and others' talks throughout, you guys have seen how this technology works. But when we applied it to our pancreatic cancer questions, as I showed on my poster yesterday, we did a bunch of analyses looking really at intratumoral spatial heterogeneity with glandular heterogeneity analysis, neighborhood analyses, ligand receptor interaction analyses, and we're really finding that we're identifying certain interactions in the tumor microenvironment that may drive chemotherapy resistance. So that's been pretty exciting. But for this story, again, you know, we took some of these same tumors that we also had geomics data, that we also had SNOOP-seq data on. And we were able to confirm through kind of prior knowledge of what genes would be enriched in what cell type compartments. You can see at the protein level and at the RNA transcript level, the architecture is very well recapitulated, which was very reassuring for us to see. And when you zoom in on a particular area of one of these SMI CosMx fields of view, you can see that the segmentation also is quite compelling, right? Just by eye, when you look at the counterstaining, that the segmentation does really quite a good job. So we were able to take this, and there was over 700,000 high-quality cells after QC filtering. And you can see on the left here, this is a UMAP with all the malignant cells in yellow and all the non-malignant cells in green. And these, of course, all have spatial coordinates, which is very important for these tumor nerve questions we're asking. And then when we actually separate out the non-malignant cells, you can see that the Schwann and nerve cells are kind of shown in this sea green color. And again, going back to our geomics data now, we took a look at these top 10 hits, and we looked for how many of these were we fortunate enough to have in the 1,000-plex panel. It turns out there was one, and that was NERP gene number 3. So we took a look at this, and the first thing we did was we looked at the observed distribution of malignant cells that express at least one copy of neural-like progenitor program gene number 3. And we basically modeled each nerve cell in the CosMx data as a Gaussian decay function. And if you sum those Gaussian decays over an entire field of view, you basically get this topology, right? So if you look here, basically the red topology is the density of nerves on an FOV. And each of these green dots is a malignant cell that is expressing at least one transcript of this gene of interest. And I think you can see here, but you can also see when you compare it to the null distribution of the FOV, the log-fold changes are positive regardless of what you set the decay radius at. And you can see, I think, even better visually, these green cells are kind of clustered around these peaks, right? There's very little in the valleys. So there's other ways to look at this, of course, but this was nice validation for this particular hit that we saw at the geomics whole transcriptome but lower spatial resolution level. It still recapitulates when you go down to the single cell level. But what about the other hits, right? So again, as Dr. Beecham and others talked about previously, this ability to go from 1,000-plex to 6,000-plex and also bring in, at that point, you can have good coverage of immunology, you can have good coverage of cancer, also good coverage of neuroscience in the same panel. This was a pretty incredible opportunity for us, and when we took a look at the literature in this review that I showed a slide from earlier, there's 38 kind of known mediators of perineural invasion. And those are all covered, as you would expect, by whole transcriptome geomics. With CosMx 1K, there's only 12 covered. Still not bad, but a little less than a third. Once you get to 6,000-plex CosMx, now you're talking over, about 80% coverage of those literature hits. So that was definitely a very exciting opportunity. So this data is very much hot off the press, but I'll show you kind of what we're doing. We haven't fully analyzed this yet, but I will tell you that every single one of our candidate hits from the geomics screen are also in the 6,000-plex. So that's very exciting to see. So here we're actually using consecutive sections on those tissue microarrays I showed you that we performed geomics on. And you can see in this is an Erica stained core from that. There's 2 yellow stars that you can probably barely make out. Those are areas of tumor-nerve interaction. And then this is what it looks like with CosMx, 6,000-plex. And if you zoom in on one of these fields of view, you can see again the nerve area is pink here. That's stained with S100B and beta-3 tubulin. And then these cancer cell glands are in green. And in this case, you can see there's not just proximity, there's heavy invasion. So in order to kind of have these two platforms be as concordant as possible, you obviously have a lot more flexibility with the single cell resolution CosMx data. We basically set the definition of a cancer cell being proximal or distal to a nerve based on a distance of 100 microns away from the 8 closest nerve cells to it in space. So now you can see in this pseudo-colored image, every dot represents a segmented cell in the 6,000-plex CosMx data. The blue dots are cells that have been determined to be proximal to a nerve, and the salmon-colored cells are those that are distant. This is just showing another image of this ultra-high-plex SMI example applied to our human pancreatic cancer specimens, and you can see, I think you can really appreciate the segmentation looks really nice, and also just the number of colored dots representing RNA transcripts is pretty incredible visually. And the QC metrics that we got out of this data was also equally surprising in a good way. We were getting well over 600 RNA counts per cell in 2 different TMAs. And again, there was no special preparation with any of these. These were archival TMAs that we had formed previously for the geomics project. And we were, out of 6,000 genes in the panel, we were able to detect, on average, about 5,000 unique genes above the limit of detection. And we were getting well over 50 to 60,000 cells from each of these TMAs. And we probably only had, with the orientation cores, we're probably talking about 10 cores per TMA. So we're very excited to really unpack this data. I'm just showing you here an example that the NanoString team put together of a geomics core with the matched cosmics core. And here you see, we define this circular overall ROI, and within it is pseudo-colored cancer cells that are proximal in purple to the nerve, which is shown in with the blue mask. And down here, you're seeing, again, really well matched. The purple segment in geomics, it has been deemed to be proximal to nerve in the cosmics data using the same definition of within 100 microns. And this is the DE analysis that, again, we haven't fully unpacked yet. Some of the hits seem to overlap, and some of the other ones are unique. So it'll be really interesting to look at how these 2 data sets really synergize. And then just to show you another visual example of this, on the left here, this is a PNI positive region. You can kind of see it with the counterstain for S100B and beta-3 tubulin in pink. And then on the right here, this is a PNI negative region where you don't see any pink nerve. And then the yellow dots represent a gene that is associated with nerve negative areas, and the cyan dots represent a gene that's associated with nerve positive areas. And I think what's striking is that you can see, one, these genes are clearly enriched in the green staining, pancytokeratin staining cancer cells, but I think you can see there's a lot more cyan dots associated with the nerves proximal, the cancer cells proximal to a nerve here, far fewer of the yellow dots. And then over here you see the opposite pattern with more of the yellow dot gene compared to the blue dots. Okay, so very briefly in like the last couple minutes, we were very interested in now taking these hits, right? We've done this comprehensive cell type specific, spatially resolved tumor nerve proximity screen. And unlike some prior work, we really, we also have CAF data, which I'm not showing today, but we really took more of a cancer-centric view on this rather than a nerve-centric view. So one of the questions, so we have 2 branches that we're unpacking. What molecular mechanisms mediate invasion, and what molecular mechanisms may mediate recruitment of nerves. So looking at the recruitment side, we kind of refined this in vitro dorsal root ganglia outgrowth assay, where we take mouse dorsal root ganglia, we dissociate them, we plate them, and what you're about to see here is a live movie taken of these neurons, which are not proliferating, but they are kind of extending their neurites, and you can see this extensive network forming over, we're about at 1 day right now, going into 2 days, and you can see how much neurite density increase there is. And then this is the segmentation that we do. Blue masks represent cell bodies, yellow represents the neurites. And let's take a look at a couple of our genes. So we ran this assay with recombinant proteins. So this is simply neurobasal media, which the nerves are pretty homeostatic in. And then we apply different concentrations of recombinant proteins based on those geomics hits, including the cosmics validated hit, NERP gene 3. So if you look at mesenchymal basal gene 2, 1 micromolar, 2 micromolar, 4 micromolar, there's really no change compared to the water control. Similarly, NERP gene 4, we see really no change. On the y-axis, this is the change in neurite length in millimeters per cell body cluster. If you look at the negative literature control, This one, the gray line is the kind of the control, which in this case is just PBS, 1% BSA. And as you increase the concentration of the recombinant protein, you start to actually see a suppression of neuritic outgrowth in the negative direction. So that was really nice to see that validate. And then what we're most excited about, again, this is the hit that we found both in geomics and cosmics, NERP gene number 3. At the nanomolar range, you can see there's a very significant stimulation of nerve outgrowth. But this is an in vitro assay, right? So we're also very interested in what happens in vivo. So we've adopted an orthotopic organoid system, which really recapitulates the architecture of pancreatic cancer very well. This is the classic KP model, but it also, in the ROSA 26 safe harbor locus, has a DCAS9 VPR, so that we can use CRISPR activation to overexpress various genes of interest. So we did this for the 10 candidate genes and also a scramble. And here you're looking at these orthotopic transplanters are off-target control, NERP gene 3, negative literature control. These are the tumors, gross on just under white light. There's a M-neon green cassette included, so you can see them fluoresce, the tumors. And then when you actually take out the tumors and stain for beta-3 tubulin in the left set of panels here, and then insulin on the right, and subtract these two signals, because islets of Langerhans, which have neuroendocrine cells, will also stain for nerve-specific beta-3 tubulin. I think it might be a little hard from where you are, but so I'll just describe it. You see, so in this case, this is a nerve recruitment assay, because we're injecting a ball of cancer cells, organoids, into a pancreas, so there's no intrinsic innervation of this ball of cancer cells. Any nerve signal that we see has to be recruited from the outside, and we saw something pretty dramatic. In the control, you see a moderate level of brown beta-3 tubulin staining that persists even when you subtract the insulin signal. The NERP gene 3, I think you can see there's brown everywhere within this tumor mass right here. And then with the negative literature control, you basically see no staining within it. And when we quantified this, in most cases we had 10 to 15 animals here. You see the beta-3 tubulin, nerve-specific minus neuroendocrine insulin signal per tumor area. For these 3 lines, the negative literature control significantly suppresses nerve recruitment compared to the off-target CRISPR control, and then NERP gene 3 significantly increases it. So we're pretty excited about this. We're trying to figure out at this point, what's happening on the receiving side, on the neurons. And with the 6,000 plex, we're also tackling, looking at things anew and also validating spatially some more of those 10 hits that I showed you. So we're quite excited about this direction. Hopefully we'll be able to contribute to therapeutics in the world of cancer neuroscience in the future. And with that, there's a lot of people I have to thank. As Dr. Beecham just mentioned, I started my lab a little less than a year ago. My postdoc mentors, Tyler Jackson, Aviv Regev, my entire amazing team, both clinically and in research, who've contributed to all this work. And then, again, I apologize, there's lots of people missing, but I put as many pictures of the NanoString folks who've been amazing collaborators through the years on here. And then, of course, our funding sources. Thank you very much.